Another disorder at this point is going to be on the dominant side. So we looked at the recessive genetic disorders. Now let's take a look at some dominant disorders. Some dominant disorders are the osteogenesis imperfecta. In osteogenesis imperfecta, this is basically when the compact bone, the shaft of long bones, is not really compact. It's actually spongy, and which cause bones to be bristle and break easily, right? Osteogenesis imperfecta lead to osteoporosis, and that osteoporosis causes bones to be prone to break, right? That in case of osteogenesis imperfecta, which actually it's a bad news, because a child, even as a child, is going to experience multiple bone break, multiple bone fractures as they grow. And also Huntington disease. Huntington disease, it's a neurodegenerative disease. That disease actually, it's not onset in the childhood. It's hardly onset in the childhood. In general, this is going to develop during early adulthood. Why this is so? This is so because this neurodegenerative disease develops progressively as an individual ages. Now, here we are going to have a normal brain with the number of brain cells that are going to be numerous here, while in an individual with Huntington disease, we're going to have that the brain cells are going to be not only abnormal, but they're not going to be abundant. And what causes this? It's an accumulation of lipids in this brain cells, and that accumulation of lipids cause this brain cells to stop working altogether and to actually die, to break down. That's why you see loss of brain cells, and that loss of brain cell is going to cause uncoordinated movement and all kind of no proper conduction of flow of nervous impulses from the brain to the organs, so that the individual is going to lose control of the organs. Now, we looked at dominant features and recessive features. But in looking at the population, not all features are so much dominant or recessive. Some are actually not very much dominant or recessive. They fall in between, right? This is the case of incomplete dominance. An example of incomplete dominance, it's actually hypercholesterolemia. Now, cholesterol is normally present in our body, and when we process cholesterol, we internalize cholesterol, right? Now, to internalize cholesterol, we need receptors on the surface of our cells so that we can internalize that cholesterol in our cells. When we don't have the receptors of cholesterol, that cholesterol being too much abundant in the blood, this is what we call hypercholesterolemia, high concentration of cholesterol in the blood. Now, in order to internalize cholesterol in our cells, we need the receptors, right? Then cholesterol is going to be internalized by a process that we call receptor-mediated endocytosis, right? When you go back to the previous chapters, we talked about receptor-mediated endocytosis, right? In a normal individual, receptor-mediated endocytosis is going to allow proper internalization of cholesterol. Now, in individuals that are heterozygous, meaning there is one allele that is being expressed, the other allele is not being expressed, these individuals are going to have half the normal amount of receptors, so that we're still going to have some level of internalization of cholesterol, but we're also going to have an unusual high amount of cholesterol in the blood, right? This is for heterozygous individual. However, individuals that are homozygous are going to have no receptors at all. That is a major problem. We're going to have a very high concentration of cholesterol in the blood, which actually can lead to death in a relatively young age in individuals, especially when they started eating things like meat, right? Because meat contains a lot of cholesterol, right? Any food of animal origin is going to contain a huge amount of cholesterol, so that when a baby starts consuming a level of food, some food that contains high amount of cholesterol, this is going to lead to hypercholesterolemia, and that hypercholesterolemia can cause high blood pressure, all kind of issues, and can lead to death. All right, so we looked at incomplete dominance. We also know that as we look at features, most features are controlled by multiple genes or interactions between genes, or interactions between genes and the environment. Okay? Now, there are some complexity in our genetics. At this point, we are going to address multiple allele traits and codominance. In multiple allele traits, a good example of multiple allele traits, it's actually our blood type. The blood type can be explained not by one single gene, right? Well, actually one gene, but multiple alleles, right? That's what we are going to see. So, an individual who is blood type A, that individual is going to have on the surface of their cells some surface markers that we call antigen A. Okay? Individuals that are type B are going to have on the surface of their red blood cells some surface markers that we call antigen B. Individuals who are AB are going to have on the surface of their cells surface markers that we call antigen A and antigen B, right? Because antigen A and antigen B are both present in this individual AB, we say that A and B are codominant. One is not dominating the other one, right? Whenever the individual has A, he expresses that A. Individual has B, he expresses that B equally, right? While individuals who are O have no A or B as surface markers on their red blood cells, we can consider that their red blood cells are naked when it comes to antigens A and B, right? So now, let's take a look at the genotypes. The genotypes of an individual who is blood type A is going to be capital I, capital A, right? Now, because you receive one set from mom and one set from dad, it can be capital I twice, right? From mom and from dad, and capital A twice from mom and from dad. The other possibility of genotype, if capital I, capital A, right? And lowercase I doesn't have lowercase a. It doesn't, right? So now, we actually see that actually we have two alleles here, but we actually have another one when we look at individuals with blood type B. This individual with blood type B are capital I, capital B, capital I, capital B. That's one possibility. The other possibility is capital I, capital B, and lowercase I. So now, for blood type, we have capital I, capital A, capital I, capital B, and lowercase I. So that's three different alleles, okay, that account for blood type. An individual who is AB is going to have capital I, capital A, and capital I, capital B, while an individual who is O is going to have lowercase I. Okay? Now, how can we determine the possibility in offspring in terms of blood type if a parent is blood type B and another parent is blood type A, right? Assuming that these parents are heterozygous for A and B, what we are going to see in the gametes, it's capital I, capital A, and lowercase I. That's for the female. For the male, we're going to have capital I, capital B, and lowercase I, right? Now, when sperms and eggs come together, you have an individual here who is AB blood type, an individual here who is B blood type, another one here who is A blood type, and another one here who is O blood type. So you understand that from two parents, dad being blood type B, mom being blood type A, they can produce children with a diversity of blood types. They can produce individual with A, children with A, children with B, children with AB, and children with O, right? So do not assume that because parents are 1A and 1B, they can only produce one kind or two kind of blood type in their offspring. Okay, they can have a diversity of blood types in their offspring, so there was no messing up, right? All right. So at this point, we're taking a look at polygenic inheritance and multifactor. Now, polygenic inheritance means that a particular feature we're going to take a look at, such as the height of an individual or the color of an individual, the skin color of an individual. These features actually are affected not only by multiple genes, but also the environment. For instance, your height depends not only on the genes you receive from mom and dad, that's one, which are multiple, but also on your lifestyle, your diet, right? If you grew up in a poor family where food was scarce, right? You are not going to grow as tall as your children that are going to be born of a mom or dad who was able to achieve high education and secure a good job and make sure that all the nutrients that are needed are at home, right? So your children are likely to be much taller for that lifestyle alone that they are going to have compared to you, right? We do see actually that in the United States, children tend to be taller compared to other children in the rest of the world, because the diet here is such that the nutritional values of food is very high. And most of the time, children are eating all the time in the United States, right? As a consequence of that, they end up being overall taller than their peers in other countries, including in Europe, right? Or in Asia, because here we eat a lot, okay? We eat a lot and food with high nutritional value, which actually cause you to see all the big, tall and big kids that we see in our community. So this is multifactorial. So the multifactorial and polygenic inheritance in general is going to describe a bell curve. What this means is that you are going to hardly have individuals who are extremely short or individuals who are extremely tall. Most individuals in the population are going to fall within a certain average, okay? So that, you have that, that bell curve on the height. This is also true for the skin color, right? Now, the more accumulation of capital letters, the darker the individual. The more accumulation of lowercase letter, the lighter the individual. This is true across all populations. Whether you consider a population of Caucasians, say in Tuscarawas County, you consider a population of African in Africa, such as in Gabon, or other countries in Africa, a population in Asia, right? They're all going to have a different shade of a color, right? So that you never have the same color across in a population. That is not possible, okay? And again, this is going to be a multifactorial feature, because if you consider two siblings, and these two siblings, one is working in an office all year long, the other one is working outside doing landscaping all year long, these two individuals are not going to have the same skin tone. That is not going to happen, right? The reason being that one is more exposed to the elements outside, and particularly the sun, right? And that individual is going to produce more melanin, okay, to protect himself from UV light, while the individual who is more indoors is not going to produce as much melanin. All right, this is also true whether you are talking about Asian, you're talking about Africans, you're talking about Europeans. This applies across. All right, other feature, it's actually when genes interact with each other. Interaction of genes can actually alter the phenotype. Now, when we consider the color of the eye, this is a good example. Now, in the color of the eyes, some people have red eyes, meaning that they have no pigment production in their eyes. And actually, the red that you see, it's actually the red of blood vessels. Okay? It's a reflection of blood vessels, because the blood vessels, right? The capillaries are very red because of, the hemoglobin they carry in the red blood cells, right? So this individual with red eyes have no pigments. Now, there is a gene that is called OCA2. That OCA2, it's a gene with a dominant form that when expressed produces brown eyes, the traditional brown eyes that most of us are going to have. Now, some of us have blue eyes. Question is, how does that happen? There are two possibilities, two reasons for producing blue eyes. One is to being homozygous recessive for OCA2, right? That's one possibility for blue eyes. The second possibility of blue eyes, it's that, well, you do have your OCA2 allele that is dominant, but you also do have a gene, HERC2, that when expressing is actually shutting off the expression of OCA2, causing this eyes to be blue, okay? In spite of the fact that you have OCA2 dominant allele that is present, right? So that is called epistasy. Epistasy, well, I'm not going to bug you with that, but know that epistasy is when the expression of one gene can be altered by the expression of another gene. Okay, you can have multiple interactions there. Then we have pleiotropic effects. In a pleiotropic effects, you have that one gene expression or lack of it is going to produce a whole array of consequences. Again, one single gene expression or lack of it is going to produce a whole array of consequences. In a case of Marfan syndrome, where actually collagen is not properly produced. Now, collagen is a protein that is typically found in large amount in connective tissues. Okay, the connective tissues are found underneath the skin, they are found in the bones, they are found in cartilages, you know, they are found in the blood. But anyway, they are found in throughout the body, right? Now that collagen is going to cause defective connective tissue, which is going to affect a whole array of organs because these connective tissues occur throughout the body, right? Our skeletal system is going to be affected, right? How so? The chest is going to be synced, right? We're going to have long limbs, long fingers, long arms, right? Scoliosis may occur. Well, not may, almost systematically. Not all the time though. Okay? Long, narrow face, loose joints. Okay? They can actually move their shoulders, their joints in ways that most of us cannot, right? The weakening of the walls of the aorta in the heart, right? Some blood vessels are going to become weak because of that lack of strength in the connective tissue. The heart itself is going to be affected. Mitral valves are going to be affected so that they can actually collapse. Enlargement of aorta, the aorta can pop up in a case of aneurysm, right? So the walls of the aorta are going to tear up. Okay, in the eyes, the lens can actually dislocate. Okay, that can be a problem, which can actually cause you to not see properly. Okay, we have other consequences in the skin, the lungs, okay, where the lungs can collapse, the skin is going to it's going to stretch, right? Be very stretchy, okay? These are some conditions that are caused by one single set of gene that is actually abnormal. That's the Marfan syndrome, a condition that is actually life-threatening, especially when the aorta is at risk of bursting, right? That can cause an individual to lose their life. Now, today, we know that genes are located on chromosomes, and we can actually read the alignment of the genes on chromosomes. There are multiple genes that have been associated with some features and associated with particular chromosomes. For instance, here, I think this is chromosome number 19, where we have the muscular dystrophy gene. We do have atherosclerosis susceptibility gene, insulin-resistant diabetes, ichthyosis, eye color. So now, these genes represented here, though, are not represented in proportion, okay? The representation here is just to tell you that this chromosome carries a number of genes which features are commonly known, right, among us, including hair color, eye color, insulin-resistant diabetes, just to mention only those, some being diseases, right, conditions, some not being conditions, okay? So nowadays, because of really health and trying to improve health, our emphasis is more and more on identifying genes that are responsible for some genetic conditions, right? Including deafness, including Alzheimer, right? Including acute myeloid leukemia, right? To mention only those, but you can see that many features that are genetic defects can be located on one single chromosome. Now, the genes are not only on autosomes, you also have genes that are carried by sex chromosomes. When we say sex chromosomes, we mean X and Y. You know that in human, a female is going to be XX, a male is going to be XY. So now it is going to be very important to pay close attention as to what genes are being carried by the X chromosomes and what genes are being carried by the Y chromosomes. Because genes that are on X do not necessarily have an equivalent on the Y. And actually, most of the time, the genes that are on the X chromosomes are going to drive the features that we see in males, right? Because, well, the X chromosome doesn't have an equivalent in a male. So whatever is carried by the X is going to be expressed, okay? While a female is going to have double X, right? So that one that carries a recessive allele can be hidden by the one carried by the dominant allele that you receive from either parents, right? So now the mom is going to produce some eggs, the dad is going to produce some sperms, so that we have offspring here. Female are going to be XX and males are going to be XY. Now you're going to have 2 sex chromosomes and 44 autosomes, right? Autosomes being chromosomes that are not sex chromosomes. All right. So if you focus on, for instance, color blindness in individuals, color blindness is carried by the X chromosome, right? But what consequence this can have in individuals. The consequence is that most of the time, color blindness is going to be expressed in males compared to females. Why? Because if a mom carries one allele for color blindness, all males are going to be colorblind regardless. Okay? But if mom is normal color with being homozygous dominant, then all males are going to have normal vision, right? So it does matter if mom is homozygous dominant for normal vision or heterozygous, so she's a carrier, right? And as a carrier, all the males that inherit the recessive allele are going to be colorblind, okay? So in sex-linked inheritance, it's going to be very important to look at, okay, well, what is the outcome in males? What is the outcome in females? Now we're going to start paying attention to that, right? So males with normal visions are going to have the dominant allele on the X. Males with color blindness are going to have recessive allele on the X. So how do we actually do the, how can we predict the outcome? So if we have a parent here, a dad who is who has a normal vision, a mom who is colorblind, mom being heterozygous, right? When we track the gametes for mom are going to be capital B and lowercase b in this instance. And now for the sperm cells, we're going to have capital B and Y, right? So now when this sperm meets this egg, we have a female who is normal vision with capital B, another female right here who is normal vision but heterozygous. Then we have a normal male here and a male who has color blindness here, right? So we can determine also the ratio, okay, of male normal vision to male abnormal vision. That ratio is going to be 50-50, right? While the females are all going to have normal vision. We can also do the pedigree for sex chromosomes, right? Sex-linked disorders, just like we did for autosomes. In this particular instance, now we're not going to say, well, what is the chance of males and females being affected is the same. This is not going to be true anymore, okay? This is not going to be true anymore. Say we have a grandfather here who is colorblind and a grandmother here who is normal vision. Their offspring is going to be, for the male, they're going to be normal vision here, okay? Now, if that normal vision here, male is having, is marrying a lady with who is colorblind, all the boys are necessarily going to be colorblind, okay? So now, when we look at the daughters here, okay, of this parents, when we look at the daughter who is actually heterozygous, marries a man who is normal vision, because mom is a carrier here, there is a possibility that in the offspring, they have a child who is colorblind, but in that case, it would only be a male. So notice that the males are the ones that are almost systematically affected, female in rare occasions, okay? So more males than females are going to be affected in this particular instance of sex-linked disorders. Unaffected sons can have parents who have normal phenotype, and for a female who have the affected features, right, they must have a husband that is unaffected, right, to have daughters that are not affected, or else they're going to all have boys that are affected. All right, so here we have the Duchenne muscular dystrophy. The Duchenne muscular dystrophy, or wasting disease, it's a condition where the muscle cells are not tightly packed together. Okay, these muscle cells that are not tightly packed together, as opposition to the muscles in mom here that are tightly packed together, these muscles are going to be so spread out that the tone of the muscle is not going to be there. As consequence of that, the individual is not going to be able to use his leg or their legs, so that they're going to end up in a wheelchair and stay in a wheelchair. And the Duchenne muscular dystrophy is actually an X-linked disorder, so which actually concludes this chapter. But before we say goodbye, I want to remind you to review the content of the chapter at the end of the chapter, or design yourself a summary for the content of the chapter, and then assess your understanding of the material, right, by answering questions at the end of the chapter or designing your own question. Have a conversation with your classmates so that you can compare your understanding, and very importantly, create a handwritten concept map, okay, for this chapter. Thank you for your attention. Bye now.